Hi everybody, I'm Keon and today I'm talking about my work on OL13B and its impact on hedgehog signaling, especially through its impact on trafficking this key protein smoothing and its role in the ubiquitination of smoothing. And so to start with, that's a lot of molecular terminology, but what is hedgehog signaling and why should we care about it? Hedgehog signaling is a key developmental signaling pathway. It's involved in a lot of processes, especially the neural tube, so determining the spinal cord development, as well as developing the hand. You can see here that when we have mutations, we get different numbers of, for example, digits. If you knock down the signal, you get almost no digits or maybe one digit. And if you overexpress it, you get many, many digits. This is similar like polydactyly in human conditions. In addition, it's involved in the neural tube. When you have problems in hedgehog signaling, you get defects in the neural tube. You can see here the brain, for example, is not properly enclosed because there's defects in neural tube closure. A common one is spina bifida, which is towards the base, but it can also cause exencephaly, which is in the head and leads to opening that is usually fatal in either in utero or is not compatible with long life. And molecularly, one of the things it does that our lab focuses on is look at the neural tube. So that's the spinal cord area that eventually develops into those spinal neurons and into the brain. Hedgehog is important in patterning that neural tube and allowing it to adopt different identities, right? Like across your spine, you need some neurons that do sensory information, some neurons which transmit information, some neurons which connect to the motor system. And hedgehog is expressed in the ventral area of the neural tube, kind of towards the stomach. And that gradient helps pattern all of these. And you don't need to know the specific identities, but it just helps pattern these neurons as you go up towards the dorsal side of the neural tube. And thus, keeping proper hedgehog signaling is key to get that proper developmental pattern.
So SSH is sonic hedgehog concentration heading.
Is SHH, which is one of the ligands that is involved in hedgehog signaling. There's a few others, but SHH is the common one that is most focused on by various researchers, since it's the most common one that functions in, for example, the neural tube. And so molecularly, hedgehog functions through this thing called the primary cilia. This is similar to, if you know, motile cilia in cells, which allow them to move or move fluid around. But it's not able to move, but it's a key sensory organelle. And so it's the presence of proteins within the cilium versus outside is strongly regulated by this area called the transition zone, which prevents what proteins can come in and exit. When hedgehog signals are off, when there's no ligand present, this receptor patch 1 accumulates in the cilia, and it then plays a role in causing smoothing, this transducer, to be constantly cycled out of the cilia, which means it can't ever build up, it can't ever activate, and we never get activation of transcription. In contrast, when hedgehog ligand is present, when sonic hedgehog is present, it can bind to patch and forces patch out of the cilia. This allows smoothing to accumulate in there, activate and bind to protein kinase A, which then triggers all these downstream transduction effects, which eventually allow this transcription factor to be active and transcribe all of these important target genes, which then allow the neural tube to top its fate, allow digits to form, and allow other forms of regulation in development. And so our lab focuses specifically on this protein OL13B. This protein is a ciliary GTPase. It plays a role in the structure of cilia as well as in proteins coming in and out of cilia constantly. It is important in a lot of the trafficking pathways. This was work that my PI did showing these same markers that I talked about earlier, some of these neural tube markers. And when you have this null mutant of OL13B where it's not active, all of these markers become dysregulated, right? You see that OL2 stretches up further, HB9 has a bigger expression domain, and patch-Z, which is a system to detect hedgehog signals, is completely different and much more spread out than this kind of very pointed induction at the bottom of the neural tube. In addition, we see that there's differences in the actual localization of some of these proteins. This is work on smoothen, which is that transducer I was talking about, which should only be in cilia when hedgehog is active. When we have this head mutant, which is the null allele of OL13B, we see that smoothen accumulates regardless of the presence of hedgehog, either in unstimulated or stimulated cases. It overaccumulates, which you can also see here on these fluorescent images, where you see smoothen in the cilia along with the ciliary marker, even when it shouldn't be necessarily. And so this then raises the question, which is what mechanism does OL13B use to actually regulate smoothen? Because one interesting thing from the last slide is that OL13B seems to do this from outside the cilia. Even when you have a construct that's defective to actually get in the cilia, which is where you would expect it to do its role, cilia are properly formed, they're the same length, they're not defective, yet smoothen is still dysregulated and still not in the cilia, or in the cilia when it shouldn't be. It's in the cell, cilia are dysregulated, but smoothen is properly regulated. So OL13B just being in the cytoplasm is sufficient to allow proper smoothen regulation, but is insufficient to allow other ciliary phenotypes. So it seems to be some alternative role of R13B. And so looking into smoothen, the first question is how does smoothen usually exit cilia in order to understand how it is defective in the case of R mutant. And in order to look at this, we looked at prior work by a lab in Boston, the Preserve lab. They looked at this SMO-UB construct, and so ubiquitin is a mark that proteins can adopt that allows them to fulfill different cellular roles or allows them to be trafficked differently or broken down. In this case, ubiquitin is used to cause proteins to exit cilia. The specific linkage allows them to be attacked for ciliary exit and either later breakdown or to just exit cilia and be in the cytoplasm. And so this lab looked at this constitutively ubiquitinated form of smoothen, where they have the ubiquitin attached at translation. So this smoothen is always ubiquitinated. And they found that this can never enter cilia, indicating that ubiquitin seems to be the pathway by which smoothen is removed from the cilia. And then they asked, how does smoothen get ubiquitinated? Because clearly it's not constitutively ubiquitinated in the actual cell. And they identified this protein, WWP1. This is a ubiquitin ligase, which means it actually attaches ubiquitin to the protein, in this case smoothen. They found that this WWP1 mutant causes basically the same phenotype as R13B henin does, where you see smoothen accumulate in the cilia even when there's no ligand present. Additionally, they looked at WWP1 itself, and they found that that only accumulates in the cilia when there's no ligand present and is removed when ligand is there. And so they theorized this model, which I'll show now, where WWP1 is actually there doing a job of marking smoothen to be removed from cilia, and that when hedgehog is on, WWP1 no longer is able to get into the cilia, and thus is no longer able to actually cause smoothen's removal, and thus smoothen can activate and perform its transduction function. But the question still remains, what role does R13B play in this process? Because it's doing something from outside the cilia that is affecting this smoothen removal process. And the key piece of evidence that our lab has that led to my work is that R13B doesn't seem to be required for that smoothen UB construct I was talking about. So you have this construct where smoothen is constitutively ubiquitinated, and even when you knock out R13B, you still see that properly removed from the cilia. And so that indicates most likely that ubiquitinated smoothen can be removed, and that R13B is acting upstream of the actual ubiquitination of smoothen. And specifically, I hypothesize that it regulates the localization of smoothen by regulating the actual protein that's able to ubiquitinate it. So if it regulates how WWP1 can get into the cilia, that then necessarily means that smoothen cannot be ubiquitinated in the first place, which would cause this phenotype that we see where smoothen accumulates in the cilia improperly. And the approach I took was this lentiviral infection approach. This is a common way that is used to express various genes of interest within cells, in this case mammalian cells, mouse cells. This is because WWP1 is not really present in the cell at sufficient quantities to sustain it endogenously. So you need this tagged construct to be overexpressed to be able to really detect it and see where it's going. And so we infected these cells and then used 3 different treatment conditions. Either they were left unstimulated, which is that no ligand case where there's no hedgehog present, or they were stimulated by that sonic hedgehog ligand, which will stimulate the hedgehog pathway, or, and I'll talk about this in a couple slides as well, they were stimulated by what's called smoothen agonist. This is particularly interesting because it works kind of downstream of patched, and thus it directly activates smoothen and allows smoothen to stay in the cilia, even when patched is also there. This will become interesting, as I'll talk about in a couple slides, because it allows us to unpick the role that R13B might be having or the WWP1 might be having. And then after all of these cells were stained and imaged by immunofluorescence to kind of detect the localization of these proteins within the cell, as opposed to markers for the cilia itself. And so moving on to the first kind of result, this is an interesting result where we have this WWP1 flag in this green channel, as well as acetylated tubulin, which is a marker to detect cilia themselves. And we see that even when we have cilia, we don't see a lot of accumulation of WWP1 in similar ways that the Preserve lab saw. A lot of these, and let me show you an example. This can be seen here. This is a zoom in of this section of the image, where we see that there's an accumulation of WWP1 near the base of that cilia, or near a portion of the cilia, but not really in the cilia in the same way that the Preserve lab indicated. You can see here that's kind of clustering, and we're not clear if this is the base or the tip, because cilia, we are not using a basal body marker to notate which side is which. But you can see that it's just kind of accumulating around the cilia, but not a clearly in the cilia phenotype that we see in the prior data. You can also see some cilia don't have it at all, even. And so This caused us to move on to look at different stimulation conditions because we were curious whether this accumulation here is a real phenotype that just looks slightly different than Pursuer's data, or is some kind of noise or some other question that isn't really answering the ciliary localization question. And to do that, we looked at these three cases I talked about earlier, which is the unstimulated, the head-shock stimulated, and the SAG stimulated. These are 60x pictures of representative cilia from these images to make it a little easier. And what we see is that this unstimulated localization into cilia seems to persist in unstimulated cases, but